So it's 12:15. We'll go ahead and get started. Hello, everyone, and welcome to today's ASBNB Breakthroughs webinar. Today's talk is entitled Everything You Wanted to Know About Pre-mRNA Splicing, but Were Afraid to Ask. I'm Eric Kenny. I'm the Data Integrity Analyst at ASBNB, and I'm very pleased to be moderating this session today. So this webinar is part of ASBNB's Breakthroughs webinar series, which is sponsored by the ASBNB journals. Specifically, today's session is brought to you by the Journal of Biological Chemistry, which has been a cornerstone in advancing biological research for over 100 years. Before we dive in, I have a quick reminder that you are welcome to submit questions at any time using the Q&A feature at the bottom of the screen. There should be a button that says Q&A. It might be behind the More button. And we'll address those during the dedicated Q&A session after the presentation. This webinar is also being recorded, and the recording will be available on the ASBNB website. But it's now my pleasure to introduce today's speaker, Dr. Aaron Hoskins. So Aaron is the Wasson Professor in Biochemistry at the University of Wisconsin-Madison. As you may have guessed from the title, work in the Hoskins lab is focused on understanding the molecular machines involved in eukaryotic mRNA processing, and in particular, the assembly and function of the spliceosome. He is also active in promoting undergraduate education and serves as the faculty advisor to the UW-ASBNB student chapter. And outside of the lab, Aaron enjoys cooking, reading, gardening, and spending time with his pets. But we're very lucky to have him speaking with us today. So without further ado, Aaron, thank you very much for joining us, and the floor is yours.
Great. Thank you, Eric. Can you all hear me okay? Yep. All right. So I'm delighted to be here today, and I'm going to give you kind of maybe a different webinar seminar than you may be used to, or a different type of talk that I normally give, and I'll get into that in just a second. So I first thought I would start off by explaining what my lab does and kind of how I became interested in the topic. So my lab studies the machines involved in eukaryotic pre-mRNA processing. And so in eukaryotes, when RNAs are made, the nascent RNA transcripts have to be processed by a number of macromolecular machines in order to generate a mature messenger RNA that can functionally encode for protein and be translated. And so RNAs need to be capped on their 5' end. These intervening sequences called introns need to be removed by pre-mRNA splicing. At the end of the transcript, you have to cut it away from the transcriptional machinery, and this is called transcript cleavage. And then the cleaved transcript needs to be polyadenylated at its 3' end. Now, I'm super interested in all these processes, and I'm a super splicing nerd. I'll say this is even my license plate. If you don't understand the joke, just hang on for 10 minutes, and this will become funnier as the talk goes on. But starting back at the beginning, so why did I get interested in this? And it really has to do with a couple papers that came out when I was a graduate student and I was looking for postdocs. So the first paper came out as a research paper from Melissa Jurica and Melissa Moore. And this involved a very early study of the spliceosome where people were able to purify highly pure spliceosome complexes from human cells and characterize the protein content by mass spectrometry. And this was quite a remarkable achievement. I'll note that Reinhard Lerman's lab was doing this at around the same time in Germany, as well as Robin Reed's lab at Harvard Medical School. But what became apparent from these studies is that there were many, many dozens, in up to several hundred proteins that were associated with the splicing machinery in human cells. And so this really intrigued me. And in particular, there were these two reviews that were published, one by Melissa Moore and Melissa Jurica, pre-mRNA splicing awash in a sea of proteins, and one by Tim Nielsen, the spliceosome, the most complex macromolecular machine in the cell, ? That really piqued my interest in the topic. Simultaneously, I was very interested in biophysics, and the postdoc project I was interested in involved applying single molecule methods to study the spliceosome, which we was kind of just emerging as this incredibly complex machine. And whether or not that was going to be possible, what really inspired me was work from Stephen Chu and Jody Puglisi's labs at Stanford. And there was a series of papers by Scott Blanchard and Ruben Gonzalez that showed that single molecule methods could be used to study the ribosome, and that's a similarly complex machine. And so I thought that this would seem to be possible. Now, once I began moving into the splicing field, and I would tell people this, I would get kind of a bunch of reactions. Um, the first thing my graduate advisor told me was, "Well, that's a pig's breakfast." Some other people said, "Huh, that's esoteric." And then the most common reaction, and one that I still get often when I tell people I work on splicing, is, "Well, we skipped that in undergrad biochemistry." Um, and so I thought a good way to use this webinar would be to provide a kind of a pedagogical lesson in the basics of RNA splicing and its importance for people who may not be so familiar with the machinery and what's going on, particularly given the past few years of research in a number of areas have implicated splicing and related processes as being really critical for understanding human gene expression in a number of human diseases. And so, I kind of admit that the way I kind of feel like when I go to conferences, especially biochemistry conferences, is that, the spliceosome is kind of second cousin to the ribosome. And so a lot of biochemists view the ribosome because they think of it as like, your protein's mom. So the ribosome is every protein's mom. Whereas the spliceosome is a little bit esoteric, and so it has this reputation of being the crazy cat lady in the nucleus that kind of does these weird things with RNA that don't really make sense, and why on earth would this ever have evolved? But nonetheless, it isn't that crazy. It actually could be understood at a structural and molecular level, and what RNA splicing enables you to do is some really spectacular things with development and gene regulation and organismal behavior. So hopefully, in about 25 minutes or so, you'll all be in love with the spliceosome. Also mentioned, because this is more of like a very high level overview, I'm not going to be talking about a lot of my research. I will mention a few things, but I will talk about work from a number of other laboratories. And I apologize if I leave anyone out. This isn't meant to be an exhaustive review, but this is just kind of a quick impression for the general audience. So beginning with splicing, what is it and how does it work? So splicing involves the removal of these intronic sequences from nascent transcripts and ligation of the flanking exons together. These intronic regions are recognized predominantly through conserved sequence motifs in the RNA. And there's going to be three of them which are essential for the splicing reaction. The 5' and 3' splice sites, which form the boundaries between the 5' exon and intron and 3' exon respectively, as well as a branch site sequence, which is going to be located closer to the 3' splice site than it is the 5' splice site. Now, all of these are going to be critical for the actual reaction that splicing, the actual splicing chemistry reaction. So that involves two sequential transesterification reactions. So in the first step, a phosphodiester bond is formed between the branch point adenosine and the 5' splice site to form a lariat intron structure, and this then releases another phosphodiester bond to free the 5' exon. So that's why this is a transesterification reaction. In the second catalytic step, the now free 3' hydroxyl group of the 5' exon attacks the phosphodiester linkage at the intron 3' exon boundary and ligates those two exons together. So this again then forms a new phosphodiester bond while releasing another one, in this case, the lariat intron product. So this is the splicing reaction, and these two chemical steps, which are pretty straightforward, are carried out by large machines called spliceosomes. So that reaction that I just showed you occurs many, many times in gene expression, particularly in humans. So human genes are very intron rich, and some of these introns can be very, very large, dwarfing the size of the exons. And so the kind of poster child for this is the human dystrophin pre-mRNA. So the dystrophin pre-mRNA, the nascent transcript, if it were to be made, completely without being spliced, would be 2 million nucleotides in length. It takes RNA polymerase about 16 hours to go from the beginning of the gene to the end of the gene. This contains 78 introns and 79 exons. However, the spliced messenger RNA is still very large, but it's only 14,000 nucleotides in size. So that means it's over 99% intron sequence that is actually transcribed by RNA polymerase and removed by splicing. So the stuff that is kept is only a very small amount of the total RNA that's transcribed, and there's only a small amount of the total gene as well. Uh, One of the reasons that people study splicing in a lot of different fields is the ability of splicing to generate transcript complexity. And kind of amazingly, this was realized kind of immediately when splicing was discovered. And so there's a very fundamental and insightful review from Wally Gilbert when splicing was discovered that kind of predicted that this would occur. And it's definitely the case. And so in alternative splicing, in a basic example, you can just choose whether or not to include a particular exon or not. So here we have a RNA with 3 exons and 2 introns. I can skip the red exon and make a messenger RNA that only contains the black and white introns, or I can include it and make the messenger RNA that includes all 3 exons at the top. And these could generate different mRNA isoforms and different proteoforms as well. Now, there's lots and lots of different types of alternative splicing. So I just gave you an example of exon skipping, but there are mutually exclusive exons, so ones which are always present in a transcript, but not together. You can have regulated intron retention. Ones that I'm particularly interested in are alternative 5' and 3' splice site selections, where basically you're choosing where in the exon to make that cleavage event. And all of these together can be used to generate many, many thousands of transcripts from our roughly 20,000 or so genes. Again, another poster child for splicing is the insect DSCAM gene. And so DSCAM stands for the Down syndrome cell adhesion molecule. And in insects, the DSCAM gene structure has this very unusual cassette exon architecture. And what happens is exon 1 is spliced to exon 2 is spliced to exon 3 is spliced to one of 12 alternates of exon 4. Then you go to exon 5, and then one and only one of 48 alternates for exon 6, and then 7 and 8, and so on. So if you multiply all these together, you quickly realize that this one DSCAM gene can generate about 38,000 potential different mRNA isoforms. It turns out flies actually need this mRNA isoform diversity and the consequential protein diversity to help wire their brains together. Human DSCAM genes do not operate this way, but nonetheless, we can have quite complex mRNA isoform generation. So one question that's kind of been pervasive in the field is alternative splicing gives you the potential for generating lots of different proteins, but are they actually being generated in cells? And the short answer is probably yes, at least to some degree. And there was a really nice paper that came out a few years ago from Joshua Kuhn, who's a colleague here at UW-Madison, and Ben Blanco and the Cox lab, where they used very sophisticated mass spectrometry methods to try to correlate alternative spliced mRNA isoforms with detection of the protein products. And in general, the data is kind of hard to go through, but they were able to find evidence for many of these, and they speculate that likely up to 75% or so or more of the mRNA isoforms generated by alternative splicing can actually generate alternative proteoforms. And the reason why those numbers don't quite match up has to do with some technical issues related to the sensitivity of mass spec. But I think the take-home message is that alternative splicing generates a lot of different mRNAs, and by and large, a lot of those are being used to generate different proteins. Splicing can have major consequences for organisms, and I'm just going to give you three examples that I find, I often think about are kind of my favorites. So one example comes from quinoa, and domestic quinoa that we eat is a sweet variety. We can't eat a non-domesticated quinoa because it contains a very bitter compound, a type of molecule called a saponin, that not only tastes bad but has hemolytic activity. Now, during the domestication of this crop, converted quinoa from a bitter variety to a sweet variety actually was due to a change in alternative splicing of a transcription factor. So a cryptic splice site became activated in a transcription factor that controlled saponin production, and this lowered production of those bitter compounds and gave rise to domestic quinoa crops. So here's another example from insects. And so this is a midge, a type of fly that lives in coastal regions. And this particular midge has a lifestyle that has to be tied uh, with the lunar cycle and actually the tides coming in on seashores and whether they're a high tide or low tide, which is then again, correlated with the phase of the moon. And it turns out that these lunar rhythms and circadian rhythms are controlled by splicing of a calcium-dependent kinase molecule. And different flies from different regions that have different tides actually splice that particular regulatory molecule in different ways and at different time points. And so here splicing is being used to control the natural rhythm and life cycle of an animal. And finally, in terms of human disease, one example from the former NIH director, Francis Collins, has to do with progeria. So progeria is a disease that results in very highly accelerated aging, and this can arise due to a missplicing event in a lamin A gene. And this missplicing event, you activate a cryptic 5' splice site in a particular exon, and this, instead of producing the lamin proteins, makes a progerin protein, and that causes this advanced aging phenotype. So in summary, you can think that splicing can have major consequences for the cell. And finally, I'm going to end with one that I think, I always personally find super interesting and kind of inspirational and kind of thinking from a biochemistry and enzymologist point of view, curious. And this has to do with a Drosophila gene called faint sausage. And the reason why it's called faint sausage is because if you mutate this gene in Drosophila embryos during embryogenesis, the trachea misdevelops and you kind of get this phenotype where it looks like sausages hanging on a string. And this phenotype is due to missplicing and changes in splicing in the embryonic and larval transcriptome. And what was really amazing to me when I saw this paper isn't that there are a lot of aberrant splicing events going on. This kind of makes sense if you're mutating a piece of the splicing machinery. But there are a lot of splicing events, in this case, 10% of the measured ones, which are occurring perfectly fine. And what this tells you is that splicing is essential, but the outcomes of splicing mutations and factors and other things can be highly transcript specific. And I find that very biochemically interesting and curious. Okay, so moving on. So we all know that splicing is important now. So what about the machine that does that? So the spliceosome is a megadalton-sized machine composed of RNA and protein components. It's about the same size as the ribosome, so 2 to 3 megadaltons or so, maybe a little bigger. Unlike ribosomes, spliceosomes are mostly protein. So ribosomes have about 70% RNA by content. Spliceosomes are mostly protein by content, but the RNAs are very, very important, and we'll get to that in a second. I always like to needle the people in translation by pointing out the spliceosome is actually, I think, a better catalyst, and so the rate acceleration for phosphodiester bond hydrolysis is quite high in splicing compared to amide bond formation in the ribosome. Spliceosomes are single turnover enzymes, and so ribosomes, you get on a message and you translate and make many, many amide bonds to complete the translation of a full protein. A spliceosome gets on a nascent transcript, it does one splicing reaction, and then it all falls apart and has to be removed from the transcript. And so that means every time you want to remove an intron, you have to go through a complicated process of assembly of the spliceosome, activation, which is formation of the spliceosome active site, catalysis, disassembly, and recycling of the components. Uh, in terms of thinking about how this machine works, sometimes people compare it to the ribosome in translation, and I don't think that's quite the right comparison. I think you're better off thinking comparisons of the splicing process with processes such as ribosome assembly, uh, uh, initiation of translation or transcription, so pick assembly, or replosome assembly. I think these are all kind of similarly type processes compared with spliceosome assembly and splicing. Splice sites, so those sites in the intron that I mentioned, are recognized by RNA, mostly the snRNAs, although not exclusively. Uh, uh, to give you an example of how this occurs, we can look at the 5' splice site and branch site. The 5' splice site is recognized initially during assembly by the very 5' end of the U1 snRNA. The U1 snRNA is a component of something called the U1 SNRP, which is a collection of proteins in RNA. That stands for small nuclear ribonucleoprotein. And the very 5' end of the snRNA base pairs with the 5' splice site. The branch site is recognized by pairing between the U2 snRNA in the U2 SNRP and the branch site sequence. And this A here becomes the nucleophile for the splicing reaction. In this particular case, the pairing has to occur in a very particular way, such that that branch point adenosine is actually bulged out of the duplex. And there's a specific pocket on a protein called HSH155, which recognizes that particular nucleotide. Ultimately, the goal of the splicing assembly process is to get a ribozyme assembled and to dock the splice sites into the ribozyme active site. So that ribozyme is going to be composed of the U2 and U6 small nuclear RNAs, and that's shown here in the second, or in the middle is the ribozyme core. And basically, we're going to dock the 5' splice site, which is going to be recognized by the U6 snRNA during catalysis, and the branch site, whose recognition I just showed you, into that ribozyme core and juxtapose them so they can actually do the catalytic reaction. All of this RNA is going to be scaffolded by a lot of proteins. And here's a protein called PRP8, which I'll talk about in a second, that you can see kind of surrounding the RNA components. So spliceosomes, in order to do that reaction, progress through a number of stages. You don't need to know the names of these stages, but just realize there are several of them. The stages are defined kind of based on two qualities. One, the cohort of proteins and RNAs that are associated with the intron, and two, the chemical state of the nascent transcript, whether or not the 5' splice site has been cleaved or not, for example, or whether or not the 5' exon has been ligated to the 3' exon. Now, kind of amazingly, over the past 10 years or so, we have structural elucidation of many, many stages along the splicing trajectory from both yeast and humans, as well as now we're getting some from a number of other organisms, including other yeast, a photosynthetic organism, and even C. elegans. And this is largely really done from pioneering work from Reinhard Lerman with Holger Stark, Kiyoshi Nagai's lab at the MRC, and Yigong Shi's lab. And so they really kind of pioneered the discovery or the structural biology of the spliceosome. And kind of as you can appreciate here, as we progress along the cycle, the structures begin looking very, very different from one another. And these structural transitions, in order to make those differences, require ATP-dependent ATPases or motor proteins that are going to be disrupting RNA, and RNA protein interactions to facilitate the structural transitions in the forward direction of splicing. And so splicing, even though the chemistry itself doesn't require ATP, nonetheless requires a large number of ATPases for the splicing reaction to occur. So to give you an idea of kind of the rearrangements, So during formation of the spliceosome active site, we go from the structure on the left to the structure on the right, and you can see it kind of first pass, they don't really look a lot like one another. And that's because there's a very large number of proteins associating and dissociating during this stage. But ultimately, you end up with a complex that completely lacks an active site to a complex that has an active site. It just doesn't have the substrates correctly docked into it yet. And that's the process of activation. For time's sake, I'm actually going to skip the next movie. But I'll just say there are excellent movies online about this, particularly from Kiyoshi Nagai's lab, from Max Wilkinson and Clemens Plaschke, and you can look up those up online. The next point I want to make is that the core machinery of the spliceosome is very well conserved. And here you're looking at the catalytic RNA core, so the ribozyme part of the spliceosome from human and yeast spliceosome structures. And you can see they're essentially identical. And so the ribozyme factor is conserved, and this was noted very early on by Dave Brown, Christine Guthrie, when they cloned the U6 gene from yeast. Moreover, it's very obvious that the splicing machinery evolved from group 2 introns or group 2 intron-like ancestor. And you can see that again in conservation of the RNA catalytic core. So this is the catalytic core from a group 2 intron. You can see the same type of core in the spliceosome. Very, very similar arrangement. And this is a picture I've stolen from a very good biochemistry textbook. Conservation goes beyond just the RNA components of the spliceosome. This also includes the proteins. So as I mentioned, the scaffold protein that surrounds the RNA is there's a very large one called PRP8. It's often called the heart of the spliceosome. It's the largest and most conserved protein in the spliceosome and one of the most conserved proteins found in eukaryotic nuclei. And it contains a number of domains kind of all stitched together. I'm going to call your attention to a reverse transcriptase domain. And this is a work from Leninger, but it's inspired by work from Nav Tor and Anna Marie Pyle, showing the amazing structural similarities of the reverse transcriptase domain from the spliceosome in PRP8 compared to those found in group 2 retrohoming reactions, to the reverse transcriptase in telomerase, those involved in HIV reverse transcription, for example, or viral RNA-dependent polymerases. So there's conserved architectures here that have been carried out or remained in place throughout evolution. This isn't to say that small proteins can't have really large impact. And so this is an example from my own lab where we studied a very small protein called FIV6 and found that if you removed it, you can actually change mRNA isoform generation. And so even really small things can have very large impacts on splicing outcomes in cells. So we're going to finish up by thinking about splicing and disease. And so splicing is highly medically relevant, and there's been a large number of papers covered about this. And one emerging topic has been thinking about spliceosomopathies. And so these are diseases which can range from blindness, developmental disorders, cancers, you name it, neurodegenerative diseases related to defects in the core splicing machinery. Cancer is often frequently cited as a spliceosomopathy, and so a key paper from Michael Seiler a few years ago found that splicing factor mutations are extraordinarily prevalent across a very, very large number of cancer types. And so a lot of people are studying this in the medical and clinical fields. So one example of this is in myelodysplastic syndrome, where a very large number of people have mutations in a splicing factor called SF3B1. And not only that, but there's a highly prevalent mutation of a specific amino acid. And so these mutations aren't just associated with these cancers, but can actually be causal for the disease. And that's been a very hot topic of research. Mutations in splicing factors are not limited to the protein components. These can also include the snRNA components. So this is a paper that came out just a few months ago on mutations in the U4 snRNA. And there's a variety of mutations here that can cause different types of developmental and other disorders, including blindness. And what was interesting here is that the mutations functionally map to different parts of the snRNA molecule, and they cause different types of disease. And so we're going to finish up thinking about, well, splicing can cause disease. How about curing disease? And so we can think about this with a disease called SMN, spinal muscular atrophy, and work done by Adrian Krainer. And there's this really pioneering paper that came out several years ago. So what's going on in SMN or spinal muscular atrophy is that humans have two copies of the survival of motor neuron gene, SMN1 and SMN2. The SMN1 gene is functional and is used to make SMN protein, which is important for actually assembling the components of spliceosomes. And mutations in this process lead to failure of correct formation of neuromuscular junctions, and that could lead to spinal muscular atrophy. The SMN2 gene is dysfunctional because it cannot splice in exon 7. So people who have spinal muscular atrophy have a mutation in the SMN1 gene that prevents protein production. Now, the SMN2 gene may not be mutated, but again, it doesn't make protein because it can't splice in exon 7. And so you can't get protein there either. So this causes spinal muscular atrophy, which is the leading genetic cause of death in infants. Now, this can be corrected, and originally the first therapy was an antisense oligonucleotide called nusinersen. Nusinersen targets the SMN2 gene to activate the splicing in of exon 7. Now, with exon 7, patients who have SMA, they still have a dysfunctional SMN gene, but now they've corrected splicing of SMN2 in a way that allows it to make protein. And so you can make SMN protein and correct the disease phenotype. I'll say amazingly small molecules can also do this. And so risdiplam is a small molecule that has a very similar function in restoring SMN2 splicing. And both the antisense oligos and the small molecules can be exquisitely sequence specific. And this is a hot topic in the research community at the moment. Finally, I'll finish up with thinking about drugging undruggable proteins. And so here, and this is pioneering work from Beverly Davidson and coworkers, they noticed that they can give a drug to a model system for the Huntington's disease. So this is the HTT mRNA. And this drug causes something called exonization, and basically causes an exon to be spliced in where there was previously no exon recognized before. And so human introns are long, and so there's regions which are cryptic-like exons that can be exonized by drugs and be spliced into transcripts. Now, if this happens, it turns out that you can degrade the Huntington's messenger RNA and make lower amounts of Huntington. And this is the basis for the drug Votoplam, which is in clinical trials. More broadly, you can think of this as a tool for drugging undruggable proteins. So let's say you have a gene that needs to be spliced, and that splice makes a protein, and this protein maybe is toxic for some reason. If you add a splicing modulator in there that causes exonization, the drug can cause an exon to be formed in that intron that's spliced in. This causes a premature termination codon, yielding no protein expression and decay of the messenger RNA by a process called nonsense-mediated decay. I'm going to skip the last part to make sure we have plenty of time for questions. And so I'm just going to end by some future challenges and directions for the field. And so there's a lot still we need to learn about splicing. And so I think the basis for splicing proofreading and fidelity is very poorly understood. It's likely involving kinetic competition between different processes. But if it's a kinetic competition, ideally you would like to know those numbers, and we don't yet. The structural basis of alternative splicing regulation is very nascent at this point. Understanding the impact of molecular foundations for when splicing happens and where. And here I'm talking about whether splicing happens co-transcriptionally or not, or if it happens in phase-separated compartments in the nucleus or not, and how those things are controlled and how much they matter. Moving from probabilistic models for alternative splicing prediction to mechanistic-based models. And what that means is we have very good tools for predicting alternative splicing, such as Splice AI, but they're based on probabilistic inferences. We want to go from there to actually molecular mechanisms. Understanding coupling between splicing and other cellular processes, especially transcription and cleavage and polyadenylation. understanding the role of RNA structures and modifications, and of course, continued development of new spliceosome targeting and correcting therapies. So with that, I'll end. I'll thank my lab who support me in all I do. We have funding from Remix Therapeutics, the Edward P. Evans Foundation, and a large majority of our funding is from NIGMS. I'm also going to plug a conference I'm sponsoring next summer in single molecule biophysics. I'm happy to answer any questions.
All right, thank you very much, Aaron. That was a really fascinating presentation. So we have about 10 minutes for questions. That was very on time. As a reminder, there's a Q&A button at the bottom of the screen you can use, or otherwise you can enter them in the chat. That's fine too. We already have some rolling in, so we'll jump right into it. The first question, this viewer asks, I have heard that pre-spliceosome assembly, U1/BP/U2 interactions can be quite dynamic and indirect, and then in parentheses, SR as a bridge. Is it known if any given U1 is interacting with many U2s simultaneously?
So so they are very dynamic, and so part of the work for that's been from my lab, but as well as other labs. There is structural data and biochemical data to support direct U1, U2 interactions, particularly in humans. I don't think anyone has ever tested whether or not you could get multiple U2 interactions with a single U1. I think it should be possible, but I think testing that has never been done. That'd be interesting because it could have implications for how you choose, how you define splice sites. But that's a really good question. I just don't think we quite know yet.
Is that difficult to test? Is that part of the barrier?
Yeah, because so you'd want to either do a very sophisticated pull down clip-seq type experiment to confirm that a single U1 is interacting with both things. Or this could be done by single molecule methods as well. But I just don't think anyone has explicitly asked that question yet.
All right, good field of research then. Next question. This viewer asks, let's say, thank you very much for the presentation. Can you please comment on the crosstalk between certain internal mRNA modifications and alternative splicing? There is emerging literature on the connection between splicing and N6-methyladenosine. Are other RNA modifications also interacting with splicing?
Yep, absolutely. And so I don't have these off the top of my head, but for example, it's well known that modifications in the nascent transcript can impact how splicing factors can engage the transcript. And so, for example, there are alternative splicing factors and even some constitutive factors that bind differently if, for example, N6A is present or not. Another example is pseudouridylation. So the snRNAs themselves have a number of modifications, including pseudouridines. And there's pretty good evidence in yeast from Yitao Yu's lab, Charles Query's lab, as well as my lab, that pseudouridine modifications, some of which can be induced during stress, can control conformational equilibria in the snRNAs. And so I think that they can directly modulate the splicing reaction in that way. So there's definitely, this is a very hot topic and emerging area. We're kind of at the stage where I think we can identify the modifications, but getting to the mechanistic consequences of them is still challenging.
All right, next question. Is there a possibility that some splicing events happen outside of the nucleus?
Highly unlikely, in my opinion. I mean, so there is, I will say a few caveats. So there's non-spliceosomal splicing. And so tRNAs, for example, can be spliced on the surface of mitochondria, and that's done by a machinery that's like independent of the spliceosome. Whether or not things can be spliced outside the nucleus, the kind of cases where this may occur if the nuclear, you can have things called nuclear budding, where basically part of the nucleus can bud off and you form this little vesicle, and there may be a lot of things trapped inside it, and this may happen in certain cancers or under certain conditions. And there, I think technically it may happen outside the nucleus, but it's just, I think, because you kind of encapsulated part of the nucleus and then it just went somewhere else. I don't think there's any evidence that you're doing um, splicing by assembling of stuff from the cytosol, like in a dendrite or something in a neuron yet. But I'd be highly skeptical, but you never know.
So if it does happen, then it's probably gone wrong, is the idea.
Yeah, I think it's in, whatever, this nuclear budding stuff, it also happens in viral replication. And so it is maybe non-traditional
Okay.
splicing.
Next question. Do you know if there is any correlation between the nuclear location of splicing and isoform determination? For
Uh,
example, is a certain isoform more likely to be produced if it is transcribed near a compartment like a nuclear speckle than if it is transcribed in a different area?
this is a very hot area of research. Uh, I don't know of particular examples off the top of my head, but there's two groups I think are really leading the way at this. One is Mitch, and I'm blanking on his last name. He's at Caltech. He developed methods called Spider and other high throughput sequencing methods that are being used to address this. The other, Mitch, um, I'm blanking on his last name. I'm sorry, Mitch. The other is Jingyi Fei at University of Chicago, who's doing microscopy-based methods. And both of them have kind of narrowed in on the idea that splicing changes in proximity to nuclear speckles. Um, so I'm very much a believer in both of their pieces of work. How this is being used by cells and how it's being regulated and the molecular explanations, I think, are still being worked out at the moment.
All right, that brings us to 12:57. So thank you so much, everyone, for the questions, and Aaron, thank you for fielding those. I just wanted to pass it back to you. Any final thoughts? Anything you would like to plug, like the conference, or anything else you want to leave us with?
Yeah, so I'll say so for the conference, this is the Physics of Life, the Nanoscale Conference at the Aspen Center for Physics, especially for people who are interested in theoretical and experimental single molecule methods, for a wide variety of systems. And so the registration and stuff is just starting up right now, and feel free to email me if you have any questions. Other than that, I hope I demystified some of splicing for you, and I hope maybe some of you now love the spliceosome and realize kind of the cool things it can do and the cool impact splicing can have on biology.
There really is a deeply fascinating amount of complexity just looking at it, even from that,
Yep.
1,000-foot view.
Yep.
Um, but anyway, so thank you so much for lending us your time and your expertise. Thank you everyone else for joining us, and please be on the lookout for future Breakthroughs webinars.
Great. Thank you. Bye.
Thanks.